Hello everybody, welcome to my channel. I'm Aisha and in today's video we are going to talk about microscopy in microbiology. So why is it important and what are the different types of microscopes? Where are these used? In which, let's say we are talking about bacteria. So what kind of microscope do we use to detect bacteria? And what do we use for viruses? Why do we use a particular microscope for a particular microorganism? We'll be focusing on all of these topics for your university exams if you're preparing for them. The questions that can be asked is like dark field microscope, electron microscopes, the principle behind them. So knowing the principle of how they work is very important, and to know what organism the particular microscope is used for is very important. Okay, so let us start with the basics of why do we need a microscope in the first place. So we are surrounded by these microorganisms and a lot of them are not visible to our naked eye, like bacteria, fungi and parasites. Most of them are present in, like, their size is in micrometers, whereas your viruses are the size of nanometers. So this is not visible to our naked eye. So obviously we require a microscope to view them. The textbook further goes on to say that fungi can be of various sizes, like they can be in micrometers, they can be in centimeters. Some parasites are visible to our naked eye because they are present in meters, whereas a lot of them are also in micrometers, millimeters. When we study about parasites in detail, we look at the various sizes that they are present in. So moving on, we know what's the requirement for a microscope now. What are the different types of microscopes? So let's just look at the names now. We'll go into detail later. So we have bright field microscope, dark field microscope. The name suggests like it is bright field. That means the field is bright and the organism inside is dark. Okay, so that gives the name. Whereas your dark field microscope is saying that the background is dark, whereas the organism that you're looking at, it is bright. Okay, so look at this word, the field. Okay, in phase contrast, that means there is some contrast. What is contrast? Contrast means that let us say we have a black background. Okay, we have a this is pink, but I'm just telling you that, imagine, okay, this is our black background. Okay, now against a black background you're able to look at the white very easily. But let us say you have a pink background and you're looking at red, it's not that good, right? So that means this is a good contrast, whereas this is a bad contrast. So that the name suggests that in a similar phase, this microscope is creating some sort of contrast so that you're able to differentiate. When we discuss about the principle of this microscope, we'll go into the detail of it. Okay, next is your fluorescence microscope. Now we know that fluorescence means there is some light that is being emitted. So we are coating the microorganism with some special material and we are putting the UV light or something, and then this thing is getting some fluorescence over this dark background, hence this name. And then electron microscope, it has something to do with the electrons. So by these names we can figure out what all of these microscopes do. Now, what are the general properties that a microscope should have? These are three main properties that a microscope should have. These properties are resolution, contrast, and magnification. Starting with what is resolution, okay? So, try to understand this. Let us say there are two distinct dots, but they are very close together. Now, the ability to tell them apart despite them being so close together is called resolution. So, let us say you're looking at the big, huge sky. You look at these stars which are light years away from you, and maybe you look at the star and it just looks like one star to your eyes. But if you start using a telescope and then you look at it further, and then it looks like, oh, these are two or three different stars. So that is the resolution power. So our human eye is limited. Our resolution, for example, let us say there are two dots that are just 0.2 micrometers apart. Now, the resolution of the human eye is 0.2 mm. So, because it is an mm and there's separation between these two dots is micrometers, our eye is not going to tell them apart because our resolution isn't that strong. But a light microscope, on the other hand, it has a resolution of 0.2 micrometers. So this light microscope is going to tell these two dots apart. Let us say we have another object like a microscope which has a resolution of 0.5 micrometers. This cannot tell these two dots apart because the resolution is higher than what is required. Okay, so I mean lower technically, but then you know what I'm trying to say, right? The values do not match. So to a resolution which doesn't match this, it is just going to look like a blur dot or one dot instead of the two separate dots that they were. So the human eye has 0.2 mm resolution. That's why we cannot see these bacteria, fungi, or parasites that are in micrometers because our human eye can only see millimeters. Next, the light microscope, because it can see in micrometers, it cannot tell nanometers apart. That's why you cannot use a light microscope for viruses. Now that makes sense, right? Like we were gonna see why are there so many different microscopes? Why can't we just stick to a light microscope and call it a day? Because a light microscope is limited to 0.2 micrometers. What if we have a virus that's in nanometers? So for that we have electron microscope that has a very high resolution, that is 0.5 nanometers. So it is able to see these literal nanometers too. Okay, so electron microscope is used for viruses. A light microscope, because it has a resolution in micrometers, you can use it for your bacteria, fungi, and parasites, and so with these other microscopes too. Okay, moving on. Your resolution is also directly proportional to the refractive index. So this is your physics. I won't go into detail, but more the refractive index, more is the resolution. So we know that oil has a greater refractive index than air. Okay, so that's why we use an oil immersion. Like when we take a slide, we put a bacteria with stain, whatever, and then we put some oil on it and then observe it under the 100x. So we use this oil because it helps in your resolution, because refractive index increases the resolution. Okay, next thing that we are going to look at is the contrast. So I've explained contrast already while I was talking about the phase contrast microscope. But again, I'll just repeat it. So a high contrast means you can separate something from the background. Like I'm using a black background right now, and you can see all of these colors because they have a high contrast against the black background. But if I start using a black color on the black background, you cannot see it. You cannot see that I was using black, right? So it will blend in a background if it is low contrast. So your microscope should have a good contrast. It should have a high contrast. So what do we use in these microscopes to increase the contrast? Okay, so we use staining. Staining is used in light microscopes. So like I said, a light microscope, light field microscope, or a bright field microscope, the background is bright and you are able to see bacteria which are stained on it. So this is why we are using stains like Gram stain or your Albert stain or your Zn stain. We look at all of these stains when we are talking about general bacteriology. So a stain is used to increase the contrast. Now all of your microscopes do not use a stain, okay? There are some special microscopes that have some special optics, like the whole architecture, what is it called? Like the whole mechanics of the microscope is made in such a way that it is special. You don't need to use a stain. So such microscopes are phase contrast microscope and dark field microscope, and these stainings are not really used. In this, the bacteria is illuminated in such a way that you can tell it differently from the background. Like in a dark field microscope, the background is completely dark, and then your bacteria is just light and you can tell it apart. We'll look at how it works, the principle of how it works. And in a phase contrast microscope, the phase of the bacterium is enhanced. Again, we'll look at it when we are looking at the principle of it, the mechanics of it. In this as well, the microscope is visualizing bacteria that is unstained. And because your bacteria, many times, like, it is light in color and it blends in with the water. Okay, so this phase contrast microscope gives the, like, for example, if your bacteria has some motion in water, it gives some properties to this motion, and that makes it different from the normal water, and it differentiates the two transparent things. So this is very interesting. We look at it when we are talking about the principles. Let us not confuse ourselves. And the next one that helps in your contrast is your fluorescence. So basically, we are, again, like I said it before, we are going to stain our bacteria with some fluorescent material, and then we are going to put a light, and your bacteria is going to be fluorescent against the background. So again, this is a type by which we are increasing the contrast. The next thing is magnification that your microscope should have. So your bacteria is in micrometers, but then when we are observing it in a microscope, it is going to magnify it, zoom it so much that it looks like it is in millimeters or in meters. Okay, so I don't need to explain what magnification is. What is important is how magnification is calculated. So if you remember how your microscope looks like, we have these, one second, we have our ocular lenses here, and then we have four lenses here, right? So it's going to multiply both of these. So these are called your objectives, and these are called your ocular, which you put on your eyes. Okay, so objective into ocular is going to be your total magnification. So these were the basic properties of a microscope. Now let us look at each microscope in detail. Okay, so starting with our bright field microscope or the light microscope. So like I said, the name says that the background is bright and the image that is formed is dark in color. That's why it's called bright field. So this is a type of contrast, right? Like you're able to observe this dark image against the light background, and I have talked about this in contrast. So the contrast here is by staining. So our bacteria or parasite or whatever, it is stained, and hence you're able to observe it. So let us look at the parts before we talk about how this thing works. So the parts are divided into three parts. Okay, they are mechanical, magnifying parts, and illuminating parts. So illuminating parts are the parts that are helping in illuminating, right? So first, let us look at the mechanical parts. There are three. First is our base, then is our C-shaped arm, then is our mechanical stage. So starting with the base, this is the base of the microscope. So the base has a light source. Now this light source can be a mirror, or it can be so your light source is obviously a part of your illuminating part. Okay, so your light source can be a mirror, or it can be an electric bulb. The ones that we have in our labs are the ones that have the electric bulbs in them. Okay, so your base holds this light source, and then it has these fine adjustment and coarse adjustment. Again, these are a part of your illuminating parts, because this will help you adjust how the image looks like. Okay, like how fine the image is. It helps you to basically sharpen the image. Okay, so the your base holds a light source, fine adjustment, and also the coarse adjustment. So all of these come in the base. Next is your C-shaped arm. This thing, okay. So this is holding your microscope together, and it will connect these ocular lenses to your objective lenses. Now your ocular and objective lenses are a part of your magnifying parts. So what is an ocular lens? Ocular lens is the one that you put your eye in, right? Like you observe it. So that's why it's called ocular lens. So the magnification of the ocular lens is 10x. If a microscope like this microscope, it has two ocular lenses, it is called as a binocular microscope. If it has one, then it's called uniocular or monocular. Next one is your objective lenses. These objective lenses are present in a revolving nose piece. And they are of different magnifications. Those are your 4x, 10x, 40x, and 100x. Based on these magnifications, we will calculate the total magnification. Like I said in the beginning, your microscopy, the basic property is magnification. And how do you calculate the total magnification? You multiply the objective and ocular. So you multiply basically what you're seeing here from what you're seeing here. So when you multiply your 10 into 4, you get 40, 100x, 400x, 1000x, and so on. So your 40x, this is called as the scanning field. So this is used in microbiology when, like for example, you take a slide, you put something on it, and you're just initially trying to focus. So you're using the scanning field to just scan where your organism is. And also it is used for larger organisms like your parasite eggs. Some parasite eggs are bigger, so you can look at them in your 40x. Next is your low power, which is you use the objective lens, which is 10x, and then you look at it. So in total, the 10x into 10x will give you 100x. This will again be used for like some bigger samples, like your stool samples, your helminthic eggs, and stuff like that. So imagine this is increasing, right? So that means here you are able to look at organisms that are bigger. Here you will be able to look at organisms that are very tiny. So next, moving on to your high power. So that means you can look at the, let's say you have bacteria, like you have Vibrio cholerae. Okay, so Vibrio has this motility. So to look at the Vibrio is comma shape, right? So you are trying to look at the motility of the bacteria, so you can look at it in the high power because this is even more focused, right? Next is your oil immersion. So in this, like I said, in resolution, your oil has a high refractive index. So the more the refractive index, the more is the resolution. What was resolution? The ability to tell two things apart, 2 points apart. So you use your oil immersion for even smaller things like bacteria. So here is when, let's say you're doing gram staining. We'll learn about this when we talk about the general bacteriology and lab diagnosis. So let's say you're gram staining some bacteria. So you look at them in this oil immersion to look at them perfectly, like these. Okay. Next is your Zn staining. This is also called as acid fast staining. Again, we'll look at this when we are talking about the general bacteriology. So what are these two held by? These are held by your C-shaped arm. So it is connecting your ocular lens to your objective lens. Next is your mechanical stage, which is this part. It has it is consisting of these clips which will help you to hold the slide in place, and then it has this little hole. So your mechanical stage has this little hole which will help the light pass through and let it come on the slide. And what else does it have? It has stage control knobs, like these here. So they'll help you move the slide horizontally, vertically, in whatever direction you like. Okay, so we've looked at all the parts. We've looked at the mechanical parts, the magnifying parts, and the illuminating parts. And illuminating parts, there's something also called as the iris diaphragm and condenser. So this is your light source, right? So when your light comes, there is something called as a condenser. So this will help you basically condense the light on the slide. It will help you focus. And then your iris diaphragm, which is present somewhere here, it will help you adjust how much light is going to be led through this condenser onto the slide. Okay. Moving on, let us look at how this works. What's the principle? Okay, so we begin with down here. Okay, so first there's this light source that we have, which could be a mirror or your electric bulb, right? So your light source is going to send your light. Let us draw the light. Okay, 1 second. We'll zoom this out. Okay, so your light source is going to send your light to the condenser. Now, what's the job of your condenser? The job of the condenser is to condense that light. Okay, so this is going to send out light like this, and then your condenser is going to condense that light onto your specimen, something like this. Okay, so now your condenser is gathering all this light and it is focusing on this specimen. Now I said that in a light microscope, the specimen is going to be stained. So imagine this specimen is stained. The specimen is going to absorb some light in itself. Okay, so again, let us go back to uh, your light beginning from here. So there's light that's going on from the light source. Your condenser is gathering that light and giving it to your specimen. Now your specimen is stained, is going to retain some light within itself, and then it is going to give out some light out to your objective lens. Okay, so basically the step that is important here to note is your specimen, right? Because it is stained, it is retaining, it is absorbing. So this is your specimen, it is absorbing some light depending on what stain is used, and the rest it is reflecting out. So that is what is creating this sort of contrast, right? And then from the objective, again it's going to focus on the ocular, and from the ocular your eye is going to observe the light. Okay, so the image is then formed in your eyes in this following way. So I hope that's easy to understand. I'm going to read the definition that's given in the textbook, and then we'll redraw this thing, okay, as we read the definition. So let us start. So the definition says that the rays emitted from the light source, let us draw it. There are rays that are emitted from the light source, pass through the iris diaphragm. So I said the iris diaphragm is somewhere near the condenser, and it helps how much light is being passed through. So it's saying the rays emitted from the light source pass through the iris diaphragm and they fall on the specimen. So they're gonna fall on the specimen. And your specimen is going to absorb some light. Okay, the light rays through the specimen are gathered by the objective. Then your specimen is also going to emit some light rays, right? So these light rays passing through the specimen are gathered by the objective, and a magnified image is formed in the objective. Now this magnified image is further magnified in your ocular lens. Okay, this image is further magnified by the ocular lens to produce a final magnified virtual image which you observe through your eye. Okay, so I hope that makes sense. I hope the diagram makes sense as well. And now let us start with the dark field microscopy. So the setup here is very similar to that of a light microscope. This is the light microscope and this is the dark field microscope diagram. So the only differences here are, first being that the condenser here has an opaque disc, somewhat like this. It looks like this. So this allows the light now doesn't go from this part, obviously. It can go only from the oblique directions. Okay. The second being the specimen here, we are not staining it. It is just naturally going to reflect the light off. So what happens here is, let us draw it here. Okay. So the light can go in oblique directions. It can't go through this, okay? So it hits the specimen obliquely. Now your specimen is unstained. It is not going to absorb the light, but it's going to reflect off almost all of its light, okay? So this gives this reflection kind of thing. It gives the specimen a bright color against a dark background because there's no light that is hitting off of here. And you can see these things, right? Let's say there's not a specimen. There's not any specimen. So there is not going to be this reflection off. So you're not going to see any brightness in your objective because the brightness that you're seeing in the objective is what you're seeing of the specimen. So let's say there is no specimen here. There is not going to be any reflection of this light. So there's nothing here. And you're just going to see a black, dark background in your objective. But because we have a specimen here, it is reflecting off some of the light. So that is the brightness that you're observing. And then again, it goes on to the ocular and it further magnifies. And that's what you observe in your eye. So I hope that you understand this. Let me read the definition so you understand it. So, only the light, the textbook says that only the light which is reflected by the specimen enters the objective lens, whereas the unreflected light does not enter the objective. As a result, the specimen is brightly illuminated, but the background appears dark. So, hence the name dark field because the field is dark and the specimen is going to appear bright. And like I said, the specimen is not stained here. It's just the natural light that it is reflecting off. So very thin specimens that you have, like bacteria, like spirochetes, are going to be observed in the dark microscopy. Here is the diagram of how spirochetes look like. They are spirally shaped bacteria and they are very thin, they are very slender, they are very flexible, and they also have a certain motility. So, the analogy for the dark microscopy is, let's say you are in a dark room, okay? You're in a dark room, everything is black. But let's say there are small dust particles in the air. Now, obviously, these dust particles are not stained, these are moving, these are very small, these are very slender. When light hits them, you look at these as bright little dots. So, that is what happens in the dark microscopy as well. Okay, so we know that our bacteria are classified based on the gram staining into positive and negative. And positive are further classified as bacilli, cocci, and negative two as bacilli and cocci. But there are some bacteria that cannot take up the gram stain because they are just so thin, or they just take up other stains. So these are classified as other or miscellaneous bacteria. So among these miscellaneous bacteria is our spirochetes. So spirochetes like Treponema pallidum, non-venereal treponematosis, Leptospira, and Borrelia are some examples of these spirochetes. So these have a corkscrew motility. Because they have a motility and because of their shape, they are observed under the dark field microscope, and this is what they appear as. There are other miscellaneous bacteria as well. For these other miscellaneous bacteria, there are different methods that you observe them under. So that's pretty much it. There is a mnemonic to remember these miscellaneous bacteria. I'll just discuss it over here. So, my spirochete, bar cox, and chlamydia. Okay, my spirochete, bar cox, chlamydia. It doesn't really make sense, but it's just easy to remember it this way. You just repeat it 5 times and you're gonna remember it. So, my is for Mycoplasma pneumoniae. It is very tiny, doesn't have a cell wall. Spiro is for spirochetes. And to remember the spirochetes, the mnemonic is blunt. Okay, BLNT, blunt. B is for Borrelia, L is for Leptospira, N is for non-venereal treponema, and T is for treponema. Okay, and ricket is for rickettsia. Bar is for Bartonella, and Cox is for Coxiella brunetti. This causes Q fever, and we'll discuss this in CVS. So I hope you understand this concept. We'll just look at this diagram at the end. Like the light microscope, in this there was no opaque disc, so the light was going in all directions. The specimen was absorbing some light, and because the light was going in all directions, your background was also appearing light and bright. Whereas here only the specimen is being hit, and that is deflecting off the light. Because the specimen was absorbing some light, here it's gonna appear a little bit dark, and also because it is stained, here the specimen is left unstained. And then the next steps are the same. The objective is going to hit the objective, and the objective will magnify it, and the ocular lens is going to further magnify it. To calculate the overall magnification, we'll multiply these both. Okay, so that's it for the dark field microscopy. We have also looked at the organism that is observed in dark field. Now let us move on to the next microscopy. Next, we are going to discuss about the phase contrast microscopy. Okay, so the problem with most of the living cells is that they are transparent. They do not absorb any light. Okay, that's why when we were using a light microscope, we were staining these cells so that they can absorb some light and they can retain that, and that's why we can look at them as some dark molecules against a bright background. But in a phase contrast microscope, we are not going to use any stains. We are not going to use any stains, and we are still going to observe these microorganisms with just light differences. So what we observed was, when light passes through, let's say a bacteria, okay, there's some light passing through. Now when it's passing through different organelles like the nucleus, the cytoplasm, the flagella, everything has a different density, right? So the light was changing its path, it was getting a little slower, the wavelength was changing. So these things are not visible to our naked eye, right? So what a phase contrast microscope does is, it will convert these invisible phase differences into your visible brightness difference, because our human eye cannot see phase differences. What we can see is the difference between brightness. So what this does is, it is going to make these changes into a differences in the brightness. Let's see how that is done. So we have a light source, obviously, and just like our dark field microscope, the condenser has this annular opaque ring thing. Okay, it has this annular diaphragm. So what this does is, when the light passes through this, imagine it's going to form a cone. Okay, so you can see this cone of light passing. Now this condenser will obviously condense the light towards the specimen. Okay, now when the light hits your specimen, there are going to be two types of light. Okay, like look at this. There is some light that is going to pass through some denser parts, the thicker or the denser parts, and that is going to be your diffracted light. Okay, and there is there's going to be some light that is going to be direct, it's going to be unchanged. You can look at it here. Some of it is passing through the denser parts of the specimen, and this is your scattered or the diffracted light, and some light is just going to be direct, it is not going to change, so this is your unchanged light. Okay, so these waves will be out of sync because this is of a different wavelength and this is of a different wavelength. So the difference in wavelength will be about 1/4. So this is a lot of physics. Just understand that your specimen, there is going to be some diffracted light and there's going to be some direct light. Now the special part about this microscope is, before the objective we have a thing called as a face plate. Now the face plate is going to be like before, and inside this objective it's going to be embedded. So this face plate is going to take this direct light. Okay, you can see that the face plate is also it also has like kind of like an annular ring. Okay, so the direct light will pass through this. Okay, the direct light is going to pass through this, and the wavelength of the direct light is going to change. So the direct light which was unchanged, it will change its phase by 1/4. Now the difference between the direct light and the diffracted light is 1 by 2. Okay, so in short, there is an annular diaphragm that is in the condenser, and it has a ring shaped opening. It will allow a hollow cone of light, and that light will reach the specimen. When the light hits the specimen, there is going to be two types of light depending on whether it's passing through highly dense areas like nucleus or cytoplasm. If it does pass through denser areas, it is going to be scattered. Okay, and it's called the diffracted light. And there's going to be some straight light which is undeviated. Now because of this phase shift, the diffracted light will be slightly delayed. Okay, and the difference between these wavelengths will be some 1/4. Okay, and the direct light is just going to go normally. Now there's going to be a face plate in your objective. So this plate will be located at the back of your objective lens, and it has a special ring too. Now this will only cause the direct light to change the wavelength, and then by the end both the diffracted light and the direct light are going to meet, and because of the difference of this wavelength, which is 1/2 now, they are going to produce a difference in the contrast of the image. Now let us say that the direct and diffracted light, if they are cancelling each other, they are going to make darker areas. If they are reinforcing each other, they are going to make brighter areas. So this is how the phase contrast microscope works. Now what is the use of this? So it is used to look at the motility of different bacteria like Vibrio. So there's this thing called bacterial growth curve. To observe how bacteria are growing, we look at them in the phase contrast microscopes. And to look at the internal cellular components like nucleus, cytoplasm, and shapes of the cell. And the main thing that you have to note in phase contrast is that the cells are not stained. We are just observing them via the phase contrast in the light. But because we cannot look at the phase contrast in the light, the microscope is going to act in such a way that it's going to make this phase contrast into a difference in brightness by the difference in wavelength and the difference in how it's going to make those wavelengths view to us. Okay, so that's it. Let's move on to the next microscope, which is our fluorescent microscope. So fluorescent microscopes are any microscope that uses the fluorescence property. The most common type of fluorescent microscope is epifluorescent microscope. This is what it looks like. Let us look at the principle and how it works. So before we go to the principle, we need to understand that there are different types of rays, right? There are gamma rays, there are X-rays, there are UV rays, then there is visible light that is visible to us. So just before this we have UV rays, which are around like 400 nanometers, and these are not visible to our naked eye. But when we take any cell, okay, when we take some cells and we dye them with some fluorescent dyes, there are different types of fluorescent dyes like acridine orange, there is your auramine phenol. So such dyes, when we put these dyes and when these dyes are exposed to these UV rays, so these UV rays will be converted from short and invisible rays to larger wavelengths, which are like visible wavelengths. So this is what these fluorescent dyes do. So we are gonna use this principle to make this microscope work. Okay, so first we are having our specimen that is colored with your fluorescent dyes, and we are gonna have a light source. Okay, so our light source will be something like a mercury lamp, and this is going to emit different sorts of wavelength. But then we have an excitation filter. Your excitation filter is only going to allow exciting wavelengths. What are exciting wavelengths? These are your wavelengths that are around 400 nanometers, that are about the UV rays. So it will only allow the UV rays which are around 400 nanometers to go through. So it's acting like a filter. Okay, so your light source is going to emit this light, and we have an excitation filter which is only going to allow the UV rays. Now we have this mirror sort of a thing. Okay, it's called as a dichromatic mirror. So it is only going to reflect these rays that are coming out onto our specimen. Now I said, when the specimen is dyed with a fluorescent dye and when it is exposed to these UV rays, it is going to convert these UV rays from short and invisible rays to longer wavelengths. So that is what is going to happen over here. Now your specimen is going to emit higher rays of higher wavelengths, and then it is going to be reflected off of here, and then it's going to reach your objective. So after your objective we have a barrier filter. This barrier filter acts as a barrier, and it will remove any damaging rays so that it doesn't hit your eye and cause damage to the eye. And then after your objective and barrier filter, it then reaches the ocular and then your eye. So I hope that makes sense. The main logic here is this thing. This is very important to understand. Okay, so what is this used for? What is fluorescent microscopy used for? We can observe different microorganisms through this, like parasites, bacteria, your helminths, and so on. So let us look at what we can observe. So there are some organisms which are called as cyclospora. They do not need any type of fluorescent dye to be observed. Like naturally, when UV light hits them, they are going to emit off the light. So these organisms, you can say, they have some sort of autofluorescence. Okay, so cyclospora, I have written it down over here, that they are a type of protozoan parasites. So let us understand this in detail, and we'll also look at the other organisms that you can observe in a fluorescent microscope which belong to parasites. Okay, so your parasites will be divided into protozoans and helminths. Okay, so protozoans are further divided depending on their shape, okay, and their motility. So amoebas, they do not have any sort of flagella or cilia for their movement. They'll just move by their pseudopodia. So we have amoeba, we have flagellates, the name says that they have flagella, we have ciliates, and we have epicomplex or sporozoa. They appear like sporozoas, okay, like spores. So the example for epicomplexa is your cyclospora and your plasmodium. Again, your plasmodium can be observed via fluorescence microscopy, but it requires a dye. Like I said, cyclospora is having natural fluorescence. That means once UV light hits them, it doesn't need any fluorescent dye. It's just gonna reflect off the light itself. But plasmodium, it needs some sort of dye, and that fluorescent dye is acridine orange. So the example for acridine orange is plasmodium, and the method is quantitative Buffy coat examination. When we move to the chapters like CVS, we'll study about plasmodium. Plasmodium vivax causes malaria, right? So when we talk about malaria lab diagnosis, we'll look at this quantitative Buffy coat examination in detail. Okay, the other organism that uses acridine orange as a dye is filarial nematodes. Now what are filarias, what are nematodes? They come under helminths. Okay, so helminths can be your platyhelminths and nematyhelminths. Your platyhelminths are your trematodes and cestodes. Trematodes are flukeworms, cestodes are tapeworms, and nematodes are roundworms. Now your roundworms can be either in the intestine or they can be somatic, like other tissues. So in your somatic nematodes, there are filarial worms. They are called filaria because they are thread-like, and they are called nematodes because they are like roundworms. So your somatic filarial nematodes, that are helminths, which are your Wuchereria branchophyri and so on, that cause filariasis. These also use acridine orange, and the method is called quantitative Buffy coat examination. So basically, acridine orange is a type of fluorescent dye, and which is used by plasmodium and filarial nematodes to be observed under a fluorescent microscope. Next, you have your auramine phenol. which is again a fluorescent dye, and the organisms that you can observe by using this dye is your Mycobacterium tuberculosis and other tubercle bacilli. So there was a mnemonic, if you remember, A B C D L M N for the gram positive bacilli. Okay, so M being Mycobacterium. So how to remember this? I had said in the previous video that bacteria are classified into gram positive and gram negative. So positive are the ones that have a mnemonic, so cocci and bacilli. Cocci have cocci in their name, like staphylococci and streptococci, and your bacilli, gram positive bacilli, they have a mnemonic which is ABCDLMN, and your gram negative cocci start with NE, that is negative, Neisseria gonorrhoeae, Neisseria meningitidis, and all of the rest organisms which do not have any mnemonic will go into your gram negative bacilli. Okay, so your Mycobacterium is a type of gram positive bacilli. Okay, and your other tubercle bacilli can also be observed by Auramine phenol. Next is a method called immunofluorescence, which we will read about in immunology, but I'm just going to give a brief overview of what happens in immunofluorescence. So because the name has immuno in it, that means we are going to use immunoglobulins. Okay, so we will take some immunoglobulins, which are your antibodies, the Y-shaped antibodies, if you remember. So they will be labeled by some fluorescent dyes, and now you will take your microorganism, let's say, and you'll put it onto this fluorescent dye thing, and then if it gets, let's say, if the antigen and antibody, there was some cell surface on antigen, let's say, on this microorganism, it will get attached to this, and because this is having fluorescent dye, when this gets attached, this is also gonna get stained. Okay, so it will help us to detect the cell surface antigens and any antibodies if they are bound to these cell surface antigens. So there are different types of immunofluorescence methods that are direct and indirect. We will look at how these work when we talk about immunology. So direct, I'll just give like an overview again. So direct means directly the antibodies are like this thing, the directly labeled. Okay, they have some fluorescent dye, and then you're putting some microorganism, let's say, on this, and it's getting attached, and your antigen and antibody are gonna be observed under the fluorescent microscope. But in indirect, we are gonna use an antibody that is not fluorescent dyed already, and it's going to react with the antigen beforehand. Now this antigen antibody complex we are gonna put on to an antibody that is labeled, so indirectly you are staining this. So these are the types of immunofluorescence, direct and indirect. It will be used to observe different bacteria like Treponema pallidum, chlamydia. We have just looked at the mnemonic for these. These are your miscellaneous bacteria. That means these do not get stained properly by gram stain, so these require other stains. The mnemonic for this was my Spiro, Ricket, Bark and Cox, chlamydia. My was for mycoplasma. Mycoplasma we already have said that it's without a cell wall. Spirochetes, we have looked at this in the dark field microscopy when we said that these thread-like organisms that have a corkscrew motility, they look something like this. These will be observed in the dark field microscopy because they are very thin and thread-like. And then your Rickettsia and Coxiella, your Rickettsia, Bartonella and Coxiella, these can be observed in the fluorescent microscopy by indirect methods, your Rickettsia and Coxiella. And then in spirochetes we have BLNT blunt. So Treponema pallidum can be observed by this fluorescent microscopy. And then other things that can be observed are your rabies virus, and your in SLE there's ANA that we are observing, and then toxoplasma. So just now we have discussed about protozoan parasites. So toxoplasma comes under protozoans in epicomplexa in sporozoa. So epicomplexa, the other name is sporozoa. So in that we have plasmodium, we have toxoplasma, and we have cyclospora. Okay, so cyclospora comes under protozoan epicomplexa. I hope that makes sense. I just briefly discussed about the classification of parasites here too, so you understand what organisms I'm actually talking about. But don't worry if you don't understand this right now, because once we get to systemic or your general parasitology, you're gonna understand this better. Okay, moving on to the next microscope and the last one, that is your electron microscope. So electron microscopy, as the name suggests, it is going to use electrons to observe the specimen. Okay, so electron microscopy was discovered by Ernst Ruska. We have discussed about this in scientists and their discoveries. So his name starts with an E. That's how you remember that he discovered electron microscope. And there are two types of electron microscopes. One is TEM, one is SEM. T is for transmission and S is for scanning. So this makes like a 3D image. It scans your entire specimen, whereas transmission electron microscopes makes a more 2D image. Okay, this is what your electron microscope looks like. It's a very complicated structure. It's huge. Let us see how it works. These are the kind of images that your electron microscope will produce. Also, your fluorescent microscope, I forgot to show you guys. This is what the images look like in a fluorescent microscope. Okay, moving on to your electron microscope. So why do we use electrons in the first place? Okay, we are using an electron gun to fire some electrons. So why are we using electrons? Because see, in your light microscope, we were using our natural light, which is your visible wavelength, which is around like 550 nanometers or something like that. Okay, so this is very limited, but when you're using shorter wavelength of light, you're gonna have a better resolution and you can observe smaller organisms like viruses, which are present in nanometers. You will not be able to observe these via your normal visible wavelengths. Okay, so electrons have around like 100,000 times shorter wavelength than your normal visible light. So this causes your specimen to have a better resolution, which is 0.2 nanometers. So one of the differences between your light microscope and your electron microscope is that the electron microscope has a better resolution of 0.2 nanometers. Your light microscope has a resolution of 0.2 micrometers. Okay, and the radiation source here is your electrons, and there it was your visible light. Again, one of the other differences is when you're using using a light microscope, the medium of travel is your air, but here we are gonna make a vacuum space. Okay, so the medium will be your vacuum, and your specimen, you cannot use a glass slide. Okay, normally when you're using your light microscope or your dark field microscope, you use a glass slide and you place your specimen on that. But here we're going to have a specimen holder that is made up of copper, and this is going to be held in a vacuum space. Okay, because your electrons can pass through your copper. Okay, so I hope that makes sense. Another thing is, everything here is going to be magnetic, because you cannot use your normal condenser or your normal objective lens, because your normal lens cannot see electrons. You need some sort of magnetic field to observe them. Again, this is a lot of physics, but just understand that the objective lens or your projector lens here is magnetic. Even your condenser is magnetic, because it's going to create some sort of a field. Okay, so when you're drawing the diagram in your exam, please mention that this is magnetic. Okay, okay, so how is this gonna work is, your electron gun is gonna fire some electrons. Now these electrons will be going through your condenser. Okay, they'll go like this on the specimen, and they're gonna hit the specimen then, and now your specimen is gonna scatter these lights even more, and then it's going to pass through these objective lens, which is magnetic again. These are not really light light rays, but if you have studied physics, then you know that electron particles have some sort of waves. So these behave like waves, and that's how it works. So your specimen will scatter more of these electrons, and it will pass through the objective, and then it's going to hit this project lens, which is further going to magnify, and then it is going to come on your photographic film. Okay, so that's how it works. You can just remember the components and then draw these lines if you are writing it in an exam. This might be asked in an exam because electron microscope is an important question. Now, let us look at how the specimen is actually made to be put in this, because the specimen that you need here is very small. You can't use a big specimen. Okay, because the specimen that is to be viewed under electron microscope should be able to maintain its structure when it is hit by these high frequency of electrons. Okay, so a very thin specimen like 20 to 100 nanometers, which is like very small, is to be made. So how is that made is, your cells are fixated, that means they are killed, and they're structured, their structure is preserved. So these are killed by using glutaraldehyde and osmium tetroxide. Then these will be dehydrated using your organic solvents like acetone and ethanol. Then they will be embedded into some plastic polymer, and then it will be made to form a hard block, like a solid block. And then after this, it's going to be sliced by an ultra microtone knife. Now this thing is going to help it maintain this really small structure, and then you're going to place it on the specimen holder, which is made up of copper. Okay, now let us look at what are the applications of an electron microscope. So it will be used to look at your different viruses. It can, like, your viruses can be in different types of specimens, right? It can be in tissue, it can be in your stool specimen. So rotavirus is present in stool, it causes diarrhea. Okay, so rotavirus is one virus that causes diarrhea, mostly in children. So I hope you understood this lesson. I'll be sharing these notes. If someone likes, please comment down below if you require these notes. So thank you so much for watching the video. Don't forget to subscribe. In the next video, we will be starting with our general bacteriology.